I'm Amanda Vazella, and this is the assay for proteolytic activity using azocasin. Some background is an enzyme is a type of protein that catalyzes reactions. Enzymes lower the activation energy of a reaction in order to speed it up. In living organisms, this plays a major role of speeding up the reactions that happen in daily life, such as metabolism reactions. It also allows for the regulation of different reaction rates. The enzyme that we used in this experiment was trypsin. Trypsin is an enzyme that is found in bacteria and the small intestines of animals. Trypsin helps with digestion of proteins that were eaten or secreted by the bacteria. The reaction catalyzed by this particular enzyme is protein hydrolysis. It cleaves after the proteins ARG and TYR. The difference between bacterial trypsin and animal trypsin is that bacterial trypsin is secreted and animal trypsin is in the small intestines. Our introduction for our question, we did at what pH does the enzyme trypsin have the highest activity? Our hypothesis was when the bacterial enzyme trypsin is placed into a 8 to 9 pH, that will be the highest enzyme activity. Our rationale for that was because trypsin originates in human/living animals' small intestines, which tend to have a basic pH of around 8 to 9. Enzyme activity will likely be highest at these pH levels. Our methods for our experimental pH variable was pH. Our experimental conditions, we had 3 replicates for each pH 5 through 10. So 3 for pH 5, 3 for pH 6, 7, like so on. Our control variables was salt concentration 0.15 moles. The temperature was 37 degrees Celsius. And then for our negative control, we left out trypsin and added 50 microliters of the reaction buffer to the solution to maintain volume. And then we had 1 negative control for each pH. Our procedure for in the centrifuge tubes, we added 450 microliters of the 0.15 molar NaCl reaction buffer, 500 microliters of the substrate azocasins, and then 50 microliters of the enzyme trypsin. After adding the enzyme, we incubated the tubes in a water bath of 37 degrees Celsius for 20 minutes. After we added the 500 microliters of TCA to stop the reaction from continuing and precipitate the undigested substrate. Then we centrifuged the tubes at 12,000 RPM for 2 minutes, and then again at 14,000 RPM for 2 minutes to separate the undigested substrate from the supernatant. Then we observed the absorbance of each tube by setting the spectrophotometer at 440 nanometers. The substrate we used was azocasin, which is a milk protein. The random addition of sulfonamide within the substrate has a positive correlation to the absorbance measurement at 440 nanometers. For our results, we got our average absorbent values at 440 nanometers. For pH 5 through 10, the standard deviation was very low, less than 0.001. And then our t-test was also very low. And then here is bar graph where you can see that for pHs 5 through 8, it rised. And then pH 8, it was at the optimum, and then decreased for 9 and 10. Our descriptive statistics were we did the 3 replicates were performed for each pH, and our p-value was less than 0.05. And then our observance values, as the pH increased, the color of the solution went from a lighter color to a darker color, and it was like an orange-red with the enzyme. And then all of them were a clear yellowish after being centrifuged. For our discussion, the experimental design, we said that the design was adequate to test the hypothesis because the enzyme activity was measured in absorbance by the spectrophotometer. And then for our conclusion, yes, the hypothesis was supported by the results. From the pH of 5 to the pH of 8, the absorbance increased and was highest at pH 8 for absorbance at 440 nanometers. And then the absorbance decreased as the pH continued to increase, proving that enzyme activity was highest at the basic pH of 8. If we had changes for this experiment, if we did it again, we would perform 3 negative controls for each pH instead of just 1. Other groups, it is debatable whether or not our results got us the best enzyme activity, as the other groups that did pH got pH 9 for the optimum, and our hypothesis stated pHs 8 to 9 were optimal, so their data does support our hypothesis. However, another group got the results that the higher pH was optimal, such as 10 to 11, and this did not support our group's results.